Hello friends, welcome to my YouTube channel Botany Girl. Now I am showing how to perform BLAST. It is mainly an alignment tool that finds local similarity between sequences. It compares query sequences like nucleotide protein to database sequence. Let's start the practical. For this you need to search NCBI BLAST on Google or any other browser. Now I am searching the website. There are different types of blast, like nucleotide blast, protein blast, blast X, T blast N. For showing you, I am choosing nucleotide blast. I have already downloaded my query sequences, and you need to paste the query sequence within this box and then click on the blast button. BLAST breaks the query sequence into smaller parts and searches matching portions in database and further extends the alignment in both directions and calculate the statistical significance. Now you can see the result. You can see how the query sequence matches with the sequence in database. Also you can find maximum score, e-value, percentage identity, accession number. E-value means expect value. Smaller the e-value, more significant the alignment. Percentage identity means how much the query sequence matches with the sequence in the database. In the graphic summary section, you can get the graphical display where the query sequence is at the top and the red bars indicate more similar sequences. In the alignment section, you can see how the query sequence is aligned with the sequence in the databases. And in the taxonomy section, you can see the taxonomy of organism from which the sequence is derived. And this tool is very helpful for identification of species, to study phylogenetic relationship, etc. I hope you like this video and please subscribe my channel and thank you.